All right, good morning, everybody. So this is about what have we learned in the last 7 or 8 years of little cherry and X disease research. Because when we started out, when I started here at WSU in the beginning of 2017, the talk was all about little cherry, little cherry, little cherry. And my predecessor, Ken Eastwell, really got this whole thing started, identifying that yes, there was little cherry virus up here, yes, it was spreading, yes, it was causing a problem. So people talked about this, the symptoms got worse and worse every year. Small, pale fruit you couldn't sell. Typical symptoms of little cherry. Now, little cherry disease caused by little cherry virus. That's how viruses are generally named, by what they cause. And it's spread by a mealybug. It doesn't move all that far, doesn't move all that fast, and it's relatively easy to control. Remove infected trees, spray for the mealybugs, your problem can be controlled. And this is really an effective way of controlling little cherry virus. The problem largely started to get under control of the orchards that had it. And like every disease, not everyone everyone gets the disease at the same time. Some people had it, some people did not. They managed to get it under decrease in little cherry disease in their orchards. But so great, problem solved. No, unfortunately not. Because as the little cherry disease and some of the orchards that were being affected by that started to be suppressed, what we started to see was the symptoms were still there. The disease was still there. And it turned out that the little cherry virus wasn't the only cause of these small pale cherries up here in Washington and Oregon as well. And really, there was another pathogen present that kept increasing in number and incidence and frequency. And that problem is X disease. And X disease is caused by a phytoplasma, which is kind of like a wall-less bacteria, that has repeatedly harmed stone fruit production, with epidemics roughly having every 50 years or so across much of North America, from the East Coast, across to the West, and up into Canada. Now the problem is both the little cherry virus and the phytoplasma symptoms on the fruit look basically the same. Unless you really know what you're looking for and want to taste the fruit, you can't tell the difference. They look the same. They regularly even fool me, and I've probably eaten and tested more of these than anyone else. So they're very similar. So that was one of the first things we learned, that these things are very similar. Old work in the past, 40, 50 years ago, said, hey, they're completely different. They don't look a bit alike. Well, it turns out what we're dealing with does actually look very similar. We learned in studying this disease as it progressed and really up and down the state, was that all commercial cherry cultivars that are being produced right now are susceptible to this pathogen. There is not a single one that is really resistant or tolerant to any degree that really matters in terms of the disease. Symptom appearance differs with cultivar to some degree, exactly what it will look like, the disease stage, how infected is it, and how long it's been infected. And one of the important things we learned, and this is a more recent discovery, is that symptom severity correlates with pathogen concentration. The more pathogen you have in the tree, and particularly in the fruit that's becoming symptomatic, the more obvious the symptoms will be. And this is not just, I have this amount of pathogen in the tree, it's where it is in the tree. Is it in the developing fruit? Because if it doesn't get to developing fruit in the right concentration, the right time of year as it's developing, you don't get as serious symptoms or you don't get symptoms at all. So you've probably seen this in looking at trees out in the orchard. They're not, in a given tree, all the fruit don't look the same. Some are more severely affected than others in a diseased tree. And that is because of the concentration that got into those developing fruit as the disease developed. And that's really one of the major problems of this disease. It doesn't hit all trees evenly. It doesn't hit even individual trees consistently across the tree. It's about where it is in the tree and how concentrated it is. This is not to say that even a low concentration causes a problem, because it can. You do lose some degree of fruit quality and size even with a low level of infection. It's just not as obvious, harder to spot. And this brings me to the next topic that really we spend a lot of time working on, is distribution in the tree. Some pathogens, some diseases spread evenly throughout the tree. It's easy to find because it's everywhere. This is not one of those pathogens. This is a pathogen that is very slow to accumulate in the tree and very unevenly distributed within an individual tree until very late stage of the infection, where there's so much pathogen there that you can't possibly miss it. Now, movement is slow. This is a very slow pathogen. It can take years after inoculation by a leafhopper to spread systemically throughout the tree, at least more than 2 years, possibly quite a lot longer than that, depending on how much pathogen got in there in the first place. because basically the more dose you have, the faster it goes. And really, where it goes in the tree, this isn't a free-flowing organism. It does move through the phloem system, throughout the tree systemically, and the phloem's basically, and this, I'm thanking Toby for this, he's good at metaphors. The phloem is basically a series of roads stretched throughout the tree. So things can go up and down roads, it can go off onto side roads like you got here. And let's see if this works, there we go. But what it doesn't do, there's some roads that don't connect, or they don't connect very closely. There's not a lot of lateral connections between the phloem that this thing can access. So it's got to go the long way around to get around a tree, which is why it's so hard to find it sometimes. You find it there, but you don't find it over there, because it's got to get all the way around to get there. It's a slow, slow process. So where it goes, depending on where it started, in other words, where did it get in, which tissue is connected to, in other words, which roads connect to each other, and how much it got there, so where can it move. And we've also started to look into whether it's a free-flowing organism, which we don't think it is. There's some degree of interaction with the host in terms of its movement. This is not a free-flowing, quickly moving thing. And this shows when you start doing inoculation experiments and what a lot of this is based on. So in this case, we put on leaf hoppers with little cages onto specific leaves. We mark the leaves they put them on, let them feed, let them die, and then went back to the plants and just let the plants grow. Then 6 months later, we went back to these little trees, these little cherry seedlings, about 3 feet high. I just cut them up, five or six sections each, right in the same place at each time. And as you can see from the P's and the N's there, there's no real pattern to it. It was scattered throughout the tree. It's a slow pattern about where it's moving in the tree, because each tree is basically a unique situation. And when you move that up to field scale, and we look at infected field trees, this is kind of the same kind of pattern repeats itself. It's very easy to find where the pathogen isn't. And this really, really complicates detection. And particularly using destructive sampling-based methods like PCR, which is still the standard for this kind of work. And again, another Tobin metaphor. It's basically, he's grinning at me, basically comes down to, you can put mouse traps all over your floor. And if you get a dead mouse in one, well, you've got a mouse problem. And if you have a bunch of empty traps, you really don't know whether you have a mouse problem or not, and especially if you keep seeing mouse droppings everywhere. So you really don't know. Absence of evidence is not evidence of absence. It's one of those really complicating things with a destructive-based method. And this is why the work done by both the dog detection project, which has been very, very promising in terms of detection. These dogs, because they're not reliant on simply sampling a piece of tissue and getting the right bit, because they can basically sense the entire tree. All these chemical sensory methods that some other WC researchers have been working on in the last couple of years are so promising, because they're not bound to the tyranny of sampling. So they're very, very promising moving ahead. So what's it doing to the tree? We know it produces small pale cherries. We know what the symptoms look like, in general. But then we start asking, well, how? We know pathogen in, disease out, but what's going on to the tree? What's going on under the hood? And we found that by combining, and this is work of my own group, Lee Calcett's group, a couple others, Lafcotte's group, using sensory, spectral, and genetic analysis, or carbon tracing analysis, all points to one thing. The phytoplasm is getting into the plant knows it's there. Can't do very much about it. It's trying to do something about it, so the plant's trying to defend itself. It's reducing photosynthesis, and basically its limited energy budget's all being spent on defense, and it's shutting down or reducing other pathways that it would normally use to produce fruit. So basically, that's why you're getting small fruit. It's slowing everything else down to try and deal with this pathogen infection. And this is kind of a common response with a lot of systemic infecting pathogens. The plant knows it's there, it just tries to deal with it. It doesn't really succeed. But this is what's causing the small, pale fruit. The other thing that really stood out in looking at this, especially now in retrospect after working on this thing for 7 years, is the symptoms that we talk about, these small pale fruit. Typical of X disease, typical of little cherry. But in the past, in outbreaks that were in California 40, 50 years ago, and even the ones here in the 1950s or older in the East Coast and in California in the 20s and 30s, they were talking about different symptoms, these leaf proliferations, this stunting, this dieback, these tree death symptoms that we're simply not seeing. And we're like, well, something's not right here. And basically what we found out through doing a lot of genetic work and mapping, because these old strains do exist, we just don't have them, is that the strain we are dealing with up here is much more mild in that it only produces the fruit symptoms. All these other characteristic symptoms that this disease or this pathogen can otherwise produce, got it. We don't have. This thing does not cause anymore, or the strain that we have doesn't, which is why it's been so hard to find and why it slid under the radar for so long. The older outbreaks, the outbreaks in California 50 years ago, much easier to deal with in some ways because they had a lot more to go on. They could see infected trees that look like with the symptoms you see over there. Easy to spot, easy to identify, easy to get rid of. What we are dealing with is a lot harder to deal with. Something it's very slow, it's very subtle in its early stages, and it's very hard to spot unless you know what you're looking for. So this kind of begs the next question is, where's it coming from? And so if you remember the talks I've been giving over the past couple of years, one of the major focuses is it coming from the wild? Is it coming from weeds? What's its host range? And we found that, unfortunately, this pathogen has a very, very wide host range, something to the order of at least 50 species across to 20 different plant families, from dandelions through to all kinds of weeds, cherry, and a whole bunch of things in between. This has a very, very broad host range. Not all hosts are the same. Not all hosts are equal. Some are marginal hosts that really aren't that significant in terms of acquisition and spread. But the most important thing with this host range is it matches the leafhopper feeding patterns, and my entomologist friends will tell you more about that later. But basically what it comes down to is this thing can get into a lot of things because the bugs feed on a lot of things. But do these matter, was the next question. And the short answer is no, not really. This, in terms of the hosts that matter, that this thing can accumulate in, that leafhoppers are likely to acquire it from in significant number and drive spread, this is a prunus problem, this is a cherry problem. The other hosts, yes, they're a good marker of something's going on, something is being spread, but by themselves, they're not a major driver of spread in as much as the trees are. So identifying and removing those infected trees is the best way of suppressing the spread of this pathogen. So the other part of where is it coming from is where is it coming from? And we did a study from 2020 through about 2023 looking to see whether it came from the wild, because the old reports from 40, 50 years ago, or even older, say, yeah, it's coming from choke trees. It's coming from the wild. It lives out there in the wild and just comes on into the orchards because the leafhoppers come to visit. And we found that that is not happening up here in either Washington or Oregon. The phytoplasms we find, the pathogens we find outside the orchard environments, so we went around a lot of orchards and went out to see what we could find, they're different. They're not ecstasies. They're not what we're finding in the orchards. So what we're finding in the orchards up here, this is an orchard-to-orchard problem. This is not a coming from the wild problem up here. Now, this isn't true in other parts of the country because we've been sampling across the country. There are parts, say, east of the Rockies, different pattern. We are seeing movement back and forth, but not here because we're dealing with different strain of the phytoplasma, probably different vectors, just different environments. So it's hard to say that it's all the same wherever you go. It's very, very different depending on the environment. So putting this all together, and this is kind of summary of what we've learned, and with some sort of emphasizing some entomology stuff that has yet to be presented and Tobin will cover, is there's a confluence between when the pathogen is accumulating in the tree, what the tree is doing, when it's likely to be acquired, when is the real risk window in terms of acquisition and spread, and it really is from basically July through September-ish. Depending on the year, this does wiggle somewhat. But that's what's all come together to show, is that there's a confluence of factors that drive the spread of this pathogen in orchards. I see people taking pictures. I'll leave that one up for a second. Cool. All right, so putting it all together, we asked the question, we like to put this one up right before I get chased out of the room, is what's driving this? Why do we have this epidemic? What blew up in our faces and gave us this disease? And it comes down to three things, really, that matter. Change in the phytoplasma strains is one of them. The changes and what the leafhoppers are doing, which Tobin will cover in a minute. And then warmer, shorter winters is driving earlier accumulation of the phytoplasma, early emergence of the vectors. It's those three factors, more so than anything, have really been driving this. And we see that on a year-to-year pattern. So the year, say 2016, we had a nice warm winter, and warm spring, early spring. 3 years later, we have a massive disease problem. So these things are cyclical, they're temporal, and you often don't see the effect of it until a couple of years later. That's me. All right.
Thank you, Scott.